So we are going to start this deep dive in a place that feels less like a fossil excavation and more like you're standing in the presence of this beautiful cosmic lie.
Right.
Imagine you are out in the Cretaceous badlands of Wyoming. You're standing in what scientists actually call the mummy zone.
Yeah, which is an evocative name.
It is, and 66 million years of history are just, layered right there beneath your feet. And normally what you find are skeletons.
That's the standard. Jagged bones, fragments turned to stone.
Mm-hmm.
That's what deep time usually leaves behind.
Bone is mineralized tissue. It's designed to last. Soft tissue isn't. It's supposed to be gone.
Absolutely gone.
But this site was different. You look down and you don't just see bone, you see a full body outline.
Wow.
It's the three-dimensional form of a duck-billed dinosaur, an Edmontosaurus annectens. And it's not an impression. It's a specimen so complete it looks like a carcass that just dried out last summer.
And that visual, right? That idea that it just air dried like some kind of, Egyptian mummy. That single impression created this huge decades-long story in paleontology.
Exactly.
We were all taught, and we taught other people, that we were looking at actual preserved dinosaur skin.
Yes. I mean, the early finds were just so unbelievable. You think back to that first famous mummy, the old Trachodon specimen found over a century ago.
AMH 5060, yeah, a class.
Yeah, when they unveiled that thing, people were just stunned. You could see the skin texture, the creases. The entire musculature seemed to be mapped out. It felt like the one perfect glimpse we had into what a dinosaur really looked like.
And these newer finds, the ones nicknamed Ed Jr. and Ed Sr.,
Yeah.
they just blew that old amazement right out of the water.
Oh, completely.
The preservation was even more exquisite. It was pushing the absolute limits of what we thought was biologically possible for organic material.
These weren't just flat impressions. They had external soft tissue details that they completely rewrote the rule book. Let's just list some of the impossible details they could see on these things.
Okay, yeah. And we're talking about features that should have decayed almost immediately. The first things to go.
Right. So first, they saw this incredibly preserved skin texture. It was made of these polygonal, almost pebble-like scales separated by these fine little grooves. That alone is super rare.
Incredibly rare.
But then you get the accessories, and this is what's revolutionary. First, a massive fleshy crest running down the midline of the neck and the trunk. On the adult specimen, this crest was huge, like 7 to 8 centimeters high.
That is a soft tissue display feature. A huge anatomical marker that would be 100% undetectable if you only had the skeleton. It changes the whole animal's profile.
Completely. And then you move over the hips and down the tail, and that fleshy crest turns into a row of these interdigitating spikes. Spikes. Up to 5 centimeters high, and they were so thin, the edges were sharp and delicate, maybe a millimeter thick in some spots, even though the base was thicker. The sheer perfection of this just seemed to confirm it.
It had to be skin.
It had to be.
And so the historical diagnosis, based on those wrinkles and that 3D shape, was pretty simple. Air mummification. The idea was the carcass just dried out super fast in some harsh, arid environment. The skin got tough like leather, and then it got buried. That process would have stopped the microbes in the water from doing their thing right away.
It's the perfect story, right? A flash-dried dinosaur, preserved forever. But like you said, that whole idea just seems to defy everything we know about chemistry, about biology, about the limits of decay over millions of years.
And that is exactly why we're doing this deep dive. Because this perfect, scaled, crested skin, this thing we thought was an undeniable portrait of life. It's not skin at all.
It's a geological trick.
It's a geological trick. A recent exhaustive analysis has shown that we were actually looking at a geological process called clay templating. It created a perfect millimeter-thin mineral ghost of the original skin.
So we thought it was biology, but it was really a taphonomic deception, a fossilization trick. The mission of this deep dive is to unpack the science that proved the original skin is gone and reveal the fast, elegant process that created this perfect mask. Okay, let's unpack this, starting with the biggest villain of all, deep time itself.
When you talk about 66 million years, the sheer scale of geologic time, you're talking about an environment where the survival of soft organic tissue is it's chemically improbable.
Right.
The proteins, the labile lipids, the whole idea that dinosaur skin could survive is what makes the mummy claim so scientifically wild.
If you leave a modern cow carcass out, decomposition is basically done in a few weeks or months. How could proteins last from the end of the Cretaceous until today?
It comes down to chemical kinetics. The molecules that make up soft tissue, collagen, keratin, all of that,
Yeah.
they're just complex biological polymers. And over these vast amounts of time, they're attacked by one of the most common and powerful molecules in the universe.
Water.
Water. It's a process called hydrolytic fragmentation.
Okay, let's get into the technicals here for a second before we hit the analogy.
For sure. So hydrolytic fragmentation is where water molecules, HEOO, basically act as little chemical scissors. They attack and break the chemical bonds, specifically the peptide bonds, that hold those long protein chains together.
So the water literally inserts itself into the bond and just snip.
Snip, cleaves it in two. So it's not just about drying out or being eaten by bacteria. It's this constant, slow, inevitable chemical assault.
An inherent thermodynamic instability.
That's the perfect term for it. And scientific models that study decay under geologic conditions, they predict incredibly rapid degradation. Keratin, which is the tough protein in skin, hair, and nails, has a projected half-life that's just a blink of an eye, geologically speaking.
So we're talking about breakdown being basically guaranteed over a few 100,000 years, maybe a couple million at the absolute extreme, definitely not 66 million.
No way. And that analogy we use is spot on. Protein breakdown is like having a sturdy chain, the polypeptide sequence, and using water to slowly dissolve the glue at every single link.
Eventually, you're just left with dust, tiny fragments and residue.
Exactly. And even in the most exceptional fossils, we expect this universal chemical process to happen. It's a rule.
This chemical inevitability, this rule, is really the foundation for all the skepticism around soft tissue claims, isn't it?
It is, and we have to draw a really clear line here. There's a big difference between these external Edmontosaurus skin claims, which we now think are solved by this clay template idea, and the separate, much older, and still very, very contentious claims of finding internal soft tissues.
Right. You're talking about the claims of finding original molecules, things like collagen, blood vessels, even bone cells, these osteocytes, deep inside the bones of dinosaurs like T-Rex or Brachylophosaurus.
Yes. Those internal claims, they often require what critics call extreme taphonomic hypotheses. The main idea proposed for their preservation isn't drying out, but an internal chemical stabilization process.
This is the Fenton reaction idea, right? The famous iron hypothesis.
That's the what?
The idea that the body basically came with its own preservative.
Precisely. The proposal is that when the animal died, iron that was released from the hemoglobin in its blood acted as this potent chemical stabilizer. This iron, in an oxidizing environment, it caused cross-linking of the surrounding protein tissues.
So the iron basically weaves this tough chemical net around the proteins, locking them in place and protecting them from that water-based breakdown for millions of years.
That's the theory. And researchers who support it often point to the presence of iron oxides, like hematite or goethite, that are found coating or infused within these proposed soft tissues. They see it as the chemical signature of this process.
Okay, so that's the argument for it, but what's the pushback?
Well, even if that cross-linking mechanism works in the short term, the long-term survival of intact, complex polypeptide sequences, it still seems highly implausible to a lot of researchers. And the biggest reason for that skepticism is the nature of the fossil itself. Fossil bone is what we call an open system.
Okay, let's unpack that. Why does open system matter so much when you're hunting for 66-million-year-old molecules?
Because an open system means the bone is porous. Even after it's buried, it's a whole network of channels and microscopic voids. It's not some impermeable, sealed vault.
It's a sponge.
It's a sponge that, when buried, becomes a highway for groundwater, dissolved minerals, and, most critically, microbes.
So it's not just a sponge, it's actually a pretty fertile little habitat, even millions of years later.
Exactly. Modern studies have shown that late Cretaceous dinosaur bone is often colonized by this unique, thriving subterranean microbiome. These are microbes that are totally different from the ones in the dirt around the bone. The bone itself is actively supporting new life.
Which has to seriously complicate any kind of chemical analysis you're trying to do.
It's a nightmare for it. If the system is constantly exchanging material, how can you ever be 100% certain that a protein fragment or a cellular structure you find isn't just contamination from modern bacteria?
Or a biofilm from microbes that colonized the bone 10 million years ago? Or even stuff that got in during the lab prep?
You can't. Not unless you have incredibly rigorous controls. And that brings us back to that analogy. Trying to find original molecules in there is like trying to find one specific grain of original salt in a 66-million-year-old jar of soup. that's been constantly refilled with new water, new minerals, and new microbes the whole time. The system is always changing.
So what's fascinating here is that the debate really boils down to scientific rigor. The critics are intensely worried about false positives,
That's the core of it.
specifically with the techniques used to identify these proteins, like immunostaining. They can sometimes mistakenly bind to these other highly stable but non-specific organic residues, things that aren't the original protein at all.
Yes, like advanced glycation end products, or AGs.
Yeah.
That's a critical point. The breakdown of proteins doesn't just leave dust. It can leave behind these extremely stubborn, stable organic breakdown products that can, in some simplified tests, look a whole lot like the target molecule.
So if your test is designed to find collagen, but it ends up binding to some other breakdown gunk that's just sort of colored or shaped similarly, you get a false positive.
You get a false positive. And the whole challenge is that the testing methods might be getting fooled by the chemical debris left behind.
So this absolute necessity for validation, for using independent lines of evidence and really robust controls, that was the intellectual engine that drove this new research on the Edmontosaurus mummies, right?
Absolutely. The researchers couldn't just assume the skin was preserved. They had to investigate the actual mechanism of its survival.
And thank goodness they did, because that investigation led them to the greatest surprise these mummies had to offer, even before they solved the preservation puzzle.
Yes.
These new finds, Ed Jr. and Ed Sr., they delivered a massive anatomical surprise that only this kind of extreme preservation could have revealed.
And that is the discovery of the hooves.
The hooves,
Eh,
this duck-billed dinosaur, which we always imagine with like a fleshy pad or simple claws, was actually equipped with these wedge-shaped, flat-bottomed, keratinized sheaths capping its 3 hind toes.
It's a hoof reptile. It sounds like something out of a fantasy novel.
It does.
It's the first one ever confirmed. The level of detail was just incredible. The biggest hoof was over 15 centimeters long, nearly twice the length of the bone underneath it. This was a feature we had no idea existed.
And it completely changes the animal's story.
Completely. It suggests Edmontosaurus functioned more like modern terrestrial ungulate, horses or deer, and was adapted for navigating soft or muddy ground.
That perfectly preserved hoof, that crest, those spikes, that level of fidelity just demanded an explanation that went beyond the simple chemical impossibility of organic survival. So how did they test the impossible?
Well, the scientific challenge it was monumental. If chemistry says soft tissue has to break down, but the fossil shows these structures survived, how do you bridge that gap?
You can't wait 66 million years.
You can't. So they had to turn the slow grind of geology into a high-speed, high-pressure lab experiment. They used a field called experimental taphonomy.
So we're essentially fast-forwarding the burial process in a lab, turning all that geologic pressure and heat into a repeatable process.
Thermal maturation, that's what it's called.
Thermal maturation.
Exactly. And it's a technique they borrowed directly from petroleum geology, where scientists simulate how crude oil is formed. They take modern animal tissues, lizard skin, feathers, things like that, and subject them to these extreme conditions. Temperatures up to 250 degrees Celsius and pressures reaching 250 bar.
Wow.
And they hold it there for about 24 hours.
Okay, 250 bar of pressure. That is insane. That's like being under 2.5 kilometers of water or buried under almost 2 miles of rock. Why is that pressure so critical?
The high pressure is absolutely essential. It's what stops the water inside the tissue from just boiling off at 250 degrees Celsius. By keeping the water in a liquid phase, they make sure that the breakdown happening is primarily that hydrolytic fragmentation we talked about.
The chemical breakdown.
The chemical breakdown, right, rather than just some simple, unnatural drying out process.
So to hit our food analogy, it's like putting a raw piece of chicken in a super pressure cooker. You're trying to replicate millions of years of geothermal heat and that crushing weight of burial, all in a single day.
And you're making sure the chicken breaks down chemically, not just because it got dehydrated. That's a perfect way to put it.
But the early experiments had a flaw, right?
They did. Early attempts often used these sealed metal capsules, and they got important results. But they were flawed because when you seal the tissue completely, all the volatile breakdown products, the gases and liquids from decay, they get trapped in there with it.
Ah, so it becomes a closed system. The very thing the critics of the internal tissue claims are worried about with the bones.
Exactly. And that closed system leads to all these unnatural chemical reactions that just don't happen in the real ground, where those volatile materials can escape. And that's why the Chicago research team pioneered this breakthrough method. sediment-encased maturation.
This is the game changer. So how does putting it in sediment change everything?
Well, they compacted the tissues and completely encased them in fine-grained sediment, usually clay.
Okay.
So to go back to our analogy, it's like burying that piece of meat in highly absorbent porous coffee grounds inside the pressure cooker.
So the coffee grounds, the porous clay, they act like a filter.
Precisely. This porous sediment acts as a geological filter. It allows the unstable, mobile molecules, the gases and liquids from protein breakdown, to physically escape into the surrounding experimental chamber. But the stable, immobile stuff like pigments, they get locked down and they stay in situ.
In their original place.
In their original place. And this approach finally gave them a truly accurate simulation of deep time.
And the results were decisive, especially for keratin.
Oh, they were. The lab experiments totally confirmed the hypothesis, the keratin protein. It broke down, it hydrolyzed, and it was chemically lost completely. It did not survive the 24 hours of simulated deep time.
The chain broke, the dust blew away. So what was left? What did survive?
The structural ghost of the tissue survived. Specifically, what they found were melanosomes,
The pigment bodies.
the tiny organelles that contain melanin and pigment. The final results were these thin, dark films made up mainly of these exposed melanosomes resting right on the sediment surface.
So the original organic building blocks were destroyed, but the highly stable pigment structures, the melanosomes, were locked in place.
It's a fundamental shift in how we think about this. It strongly suggests that keratinous structures, the dino's skin, its spikes, even fossil feathers, they don't survive as preserved protein. Their structure survives because the extremely stable pigment molecules and maybe some calcium phosphate remnants were originally held inside that now lost keratin matrix.
The information is preserved, but the original material is gone. So this was validated in the lab. Now they had to point this tech at the actual mummy.
They did. They took extremely thin sections of the Edmontosaurus tail spike and the hooves, and they hit them with everything. Micro-CT scans for structure, X-ray diffraction to see the mineral composition, and electron microscopy to see the ultrastructure. They weren't looking for organic material anymore. They were hunting for a mineral ghost.
And that analysis of the mummy itself, it confirmed that this external preservation was totally different from the bone underneath.
A completely different process. The skeleton had gone through typical permineralization, that slow replacement process. But the crest, the scales, the hoof, that external layer was something else entirely.
The sectional analysis showed that the material was shockingly thin, right?
Less than a single millimeter thick. And crucially, the material was not carbonaceous, which would suggest compressed soft tissue. It was purely mineral.
Mineral mask. So the old idea that the dinosaur dried out in the sun like jerky before it got buried, that's definitively out. Dead.
Completely overturned. If it had been dried tissue, you'd expect to see thicker carbonized remains. This wasn't that.
So the researchers, the team from the University of Chicago, they concluded this spectacular preservation was due to a geological process called clay templating.
Clay templating. And what's so groundbreaking here is that they applied this mechanism, which we really only knew from deep-sea, low-oxygen environments, to a high-energy terrestrial setting, like a riverbed.
Which is wild. So this mechanism allowed the surface texture to be preserved, even though the Edmontosaurus died in a fast-moving river system. Okay, let's walk through the exact steps of this clay template mummification.
It's a remarkably fast and elegant process.
Yeah.
It happens in 4 main steps. Step 1, rapid burial.
Gotta get it buried fast.
Super fast. The evidence in the rock's mud-clasts conglomerates, it all points to high-energy water transport. The carcass is swept up and quickly engulfed in wet, porous sediment before decomposition could just obliterate the external features.
Okay, so the body is intact, skin and all, and suddenly it's buried in wet, silty dirt. And then decay starts immediately.
Immediately. Which leads to step 2, biofilm formation. As that soft tissue starts to break down in the wet sediment, a thin microbial biofilm rapidly coats the entire surface of the integument.
The skin, the hooves, the spikes, all of it.
All of it. And this thin layer of microbial gunk is the catalyst for the whole templating process.
So the bacteria start eating, but in doing so, they create this new sticky outer layer that becomes the staging ground for what comes next.
Exactly. Which brings us to step 3, electrostatic attraction, or my favorite term for it, taphonomic tanning.
Taphonomic tanning, I love that.
This is the chemical core of the discovery. The decaying tissue with that microbial biofilm on it creates these localized chemical conditions that electrostatically attract clay particles right out of the surrounding wet dirt.
Wait, how does dying tissue become a magnet for clay? That sounds like magic.
It's just chemistry. So clay particles like kaolinite and elite are aluminosilicate minerals. They often carry a negative charge. The decaying organic material, with its shifting pH and chemical byproducts, acts to strongly bind these aluminum-rich clay particles. The clay is literally pulled out of the sediment matrix, and it rapidly forms this continuous, thin, stable coating right on the tissue surface.
So the ELI7 version is, the dying tissue's microbial layer acts like a chemical magnet, pulling the aluminum-rich clay out of the surrounding dirt to create a dense mineral shield.
That's it. And taphonomic tanning is perfect because it's chemically fixing the surface, just like tanning leather.
But with clay instead of chemicals.
Right. And critically, these clay minerals are famous for their ability to stop decay. The high aluminum concentration physically damages bacterial cell walls, so it limits bacterial growth. It provides this temporary protective stabilization right on the surface.
So the clay isn't just providing the template, it's actively slowing down the very process that would destroy the structure it's trying to record.
Precisely. And that leads to the final step, step 4. The ghost rises.
Love it.
The thin clay layer has now faithfully recorded the perfect three-dimensional surface morphology of the skin, the scales, the spikes, the hooves, down to the smallest detail. And then the original organic soft tissue underneath that thin mineral shell completely breaks down.
It just dissolves and gets carried away by the groundwater.
Carried away in solution. And that clay layer survives, locked in place, creating a perfect millimeter-thin cast of what was there. The dinosaur's skin did not survive. Its perfect mineral mold did.
That is an enormous reversal of decades of paleontological thought.
It completely flips the air-drying hypothesis on its head. This preservation wasn't slow and dry. It was fast, wet, and chemically induced. It was probably complete within weeks or months after the dinosaur got buried.
So here's where it gets really interesting, because this isn't just a dinosaur story. This fundamentally changes how we have to assess any fossil that's claimed to have preserved soft tissue.
It does.
And it specifically puts those claims about the internal soft tissues back under a very critical microscope.
It really does. I mean, think about it. If the external thick, robust skin of this Edmontosaurus failed to survive 66 million years and had to be replaced by a clay template, it provides overwhelming contextual evidence that smaller, more delicate organic materials like blood vessels or collagen fragments inside a porous bone are highly, highly unlikely to have survived that same period.
It's the counter-validation. The robust external structure confirms that the rule of hydrolytic fragmentation, it holds true, even in these exceptional cases.
Right.
So it suggests that any internal structure that's found is much more likely to be one of three things: a highly stable organic breakdown product,
A replacement material like a mineral cast,
or contamination.
It's the difference between a durable cast and a fragile original. And the Edmontosaurus mummy shows us that nature, over deep time, almost always prefers the cast.
The scientific consequences of confirming this they're massive. It affects how we think about the durability of molecules, it rewrites the dinosaur lookbook, and it changes how we should be looking for ancient life, even on other planets.
It really emphasizes the sheer, kind of unassuming power of clay minerals. We've known clay slows down decay, but now we know it can actively create the mineral template. It attacks the microbes and stabilizes the architecture just long enough for the aluminosilicates to lock the form into place.
And this finding puts enormous pressure back on those contentious claims about internal soft tissues, the T-Rex collagen and vessels. If the clay template is the truth for external preservation, what does that mean for the Fenton reaction hypothesis for internal preservation?
It just highlights the absolute need for methodological rigor. The Edmontosaurus study used validation. They did the thermal maturation experiments to confirm that keratin is chemically lost before they even looked at the fossil.
Right.
And critics of the internal tissue claims argue that too much of that research relies on just replicating methods that might be prone to false positives, like some of those immunostaining techniques.
So the lesson here is about intellectual honesty in the lab. If you are going to make a claim as extraordinary as molecular survival over 66 million years, you have to rule out every other possibility.
Every single one. To avoid the possibility that just some generic browning gunk from organic decay is fooling your tests, researchers have suggested these crucial controls. For instance, testing tissues that never contain the protein you're looking for.
Like a plant leaf.
Exactly.
Yeah.
Take some plant tissue or muscle tissue and run it through your anti-keratin or anti-collagen antibody tests.
And if your test for keratin also lights up a piece of lettuce, then your test is fundamentally flawed. The result is meaningless.
It's meaningless. And this Edmontosaurus study provides the clean, structural proof that chemical instability wins out over deep time. It suggests that iron stabilization, even if it happened, likely wasn't enough to preserve complex polymers for tens of millions of years.
And beyond all the arguments in the lab, the clay template gave us concrete, undeniable information that is rewriting the dinosaur lookbook for Edmontosaurus.
Oh, absolutely. The fidelity of that clay template was so high that it allowed them to reconstruct the animal with a level of certainty we've never had. We know for sure it had that massive fleshy crest. We know it had that distinct interdigitating spike row along its spine.
And most importantly, the hooves.
The hooves on the hind feet.
The existence of hooves just totally changes the paleobiological story. It moves Edmontosaurus away from being some generalized duck-billed animal and firmly into the role of a dedicated terrestrial ungulated hoofed mammal analog.
They were built for navigating soft, maybe difficult, terrain. That's a functional detail we never would have known without this template.
And this realization that an elegant mineral replacement process can capture this kind of ephemeral detail in a high-energy terrestrial environment that has profound implications for the field of astrobiology.
It's huge for astrobiology.
So how does a dinosaur mummy in Wyoming help us find microbial life on Mars?
It changes our search strategy. Historically, when we're looking for ancient life, whether on early Earth or on other planets, we've focused on trying to find preserved organic molecules. But organics degrade really fast, especially on a world like Mars that's constantly being bathed in surface radiation.
Right, the harsh Martian environment would have just fried any exposed organic molecules eons ago.
Exactly. But the clay templating mechanism drastically expands the kinds of environments we should be targeting.
Yeah.
Clay-rich deposits, ancient riverbeds and lake bottoms rich in clay, are now primary targets for missions like ExoMars and Mars 2020. They are hunting in the exact environments we now know are perfect for this rapid templating process.
So even if the original Martian organic molecules were all destroyed, the abundant clay on Mars may have rapidly formed a mineral template that preserved the three-dimensional morphology.
Of microbial biofilms, or maybe even small, simple Martian life forms. The clay acts like a geological camera. It takes a perfect snapshot that can survive for billions of years, even after the fragile subject is long gone.
So we shift the focus from hunting for fragile, ancient molecules to hunting for these elegant, sturdy mineral molds of life.
It lowers the bar for what can be preserved, but raises the bar for the scientific rigor you need to prove it.
It's such a humbling reminder that deep time ensures the loss of complexity. We thought we needed these extreme, perfect, low-temperature, anoxic conditions to preserve dinosaur soft tissue for 66 million years.
But the truth is simpler, it's faster, and it's far more elegant. All you needed was a flash flood to bury the carcass, a thin layer of biofilm to act as a chemical magnet for the clay, and the universal laws of chemistry and geology to create that stable coating. The skin didn't survive, but its perfect structural ghost did.
So what does this all mean? The takeaway is that this dinosaur mummy was never a biological time capsule. It was an exquisite geological mass created by a rapid chemical reaction. And now, armed with this knowledge of clay templating and taphonomic tanning, you have to wonder, what other spectacular fossils that paleontologists are studying right now, from feathers to ancient invertebrates, are actually just perfectly disguised layers of clay-rich dirt? The history of life isn't written in the organic material that survives, but in the perfect ghostly replacements that the rocks provide.